Hello, fellow cultivators of curiosity, and welcome to the Breeding Bites NAPB Education Podcast. I'm your host today, Lance Merrick, and I'm joined with co-host Charlie Zerla. And today we will guide you through the captivating universe where seeds, genes, and everything in between converge. Whether you're out in the field, orchestrating a symphony of pollen in the nursery, or decoding the secrets in the genetics lab, you've just stepped into the epicenter of scientific discovery. In today's episode, we have Jim Holland to talk about his successful career as a corn geneticist and plant breeder. So buckle up and join us as we cultivate some captivating conversations.
Awesome. Thanks for that introduction, Lance. Yeah, I'm not normally a part of this podcast, so thanks for the introduction, Lance. For anybody who may not know me, my name's Charlie Zilla. I work with Lance at our Corteva facility in Windfall, Indiana. I'm the soybean breeder there along with Lance. Have the pleasure to work with him on a day-to-day basis. But anyways, we're not here to talk about me. I have the pleasure of introducing Jim Holland. I was Jim's graduate student many moons ago, from 2009, 2013. Did my master's thesis and PhD dissertation in corn breeding and research with Jim Holland. So Jim, I will cut myself off here. Can you just tell us a little about what you do now? What your position is at North Carolina State University, and we'll go from there.
Okay, thanks. Let me make the most important clarification. I work for USDA.
Yes.
I'm a research geneticist with USDA Agriculture Research Service. I'm located at NC State University. So what that means is I'm like any other USDA federal employee. I have a project plan that gets approved by USDA. We're basically our research is funded kind of to the level of a particular crop. At least in my case, I have to work on maize, and it has to be some kind of genetic improvement that's relevant to farmers in the U.S. So that's where USDA gives you sort of that level of direction, and then it's up to us as USDA researchers to come up with a research project plan. And then the interaction with like North Carolina State University is that I happen to be located on the campus. And that's pretty nice. In fact, this office I'm sitting in is at a university building, but I'm not a university employee, and I co-advise graduate students and things like that, but that's all done in collaboration with regular NC State faculty members.
Awesome. So maybe, as I listen to some of the other podcasts that Lance and others have put together in some of this different series, I part of the point of these podcasts is people just want to know, where did people get to this point in their careers, especially for, aspiring undergraduate students, graduate students, folks like that. And I know your history. I would call it probably a somewhat untraditional path from when you started your undergraduate work and forward. So maybe give me the full Jim Holland life story. Where are you from originally? Where'd you go to undergrad? And then how'd you get to where you're at today?
Okay.
Leave no detail out.
I'll leave a few details out.
Okay. All right.
It's not that interesting. But I was born very close to Boston, Massachusetts. But I didn't live there very long, probably less than a year or so. And then my family moved to the West Coast. So my dad was a Navy officer. He was a submariner. So we moved every 2 or 3 years, and we moved to places that were on the ocean. So I was a coastal kid. So I was either in California or on the East Coast, or a couple really nice years we were in Hawaii. So yeah, I did not grow up in agriculture. I think that's what you mean by non-traditional. I didn't grow up on a farm. I didn't know any farmers. My dad's from Iowa, actually. But maybe he joined the Navy to see something outside of Iowa. I don't know. I never really asked him why, but I have a lot of cousins who live in Iowa. I have aunt and uncle and cousins. So we would always stop there, and when we were driving from coast to coast, we would stop in Iowa and visit the family there. But even though they lived in Iowa, none of them were actually farmers either. So I didn't have any experience with agricultural production in any way. I went to college. I was very interested in science. I was a nerd. Well, still am. I'm a nerd. And, but you know, when I went to college, I wanted to learn science. I was a biology major, and I sort of thought, wow, it would be fun to do research. I didn't really know, and again, nobody in my family was a scientist either. So this was all like, I think maybe this is nice to have these podcasts. They didn't exist when I was a kid. Right? You just had no idea like what it meant to be a scientist, other than kind of sounded cool. And, I like biology. I wanted to do something though that was maybe more applied and, would have some outcome you could see that would be beneficial. And, when I was in high school, there were bad famines in the world, and, world food production was, looked like it was on a bit of a shaky ground. And so, part of my thinking was, well, maybe, I like genetics, I like biology, like, could I have a job where I do that sort of stuff, but maybe would have some beneficial outcome for food production. So that's how I ended up looking for plant breeding before I knew what plant breeding was. So again, I didn't go to an ag school. I went to I was at a university where everybody else who was a biology major almost was going to become a doctor. They were all pre-meds. I was interested in plants. There were basically no botany classes. Like it was like really fish out of water there in some sense. But it was really good fundamental background education and biology, and just getting good college background was, that was really helpful for me in that sense. But I learned about plant breeding back in the old days. Again, no internet. This is the 80s, right? So you'd go to the library, and there were these big books that would have a description of graduate departments at different universities in the US. So there would be this giant thick book, and you could flip through it. And so I was looking through, like, I would look at biochemistry, and I was like, I don't think I really want to do that. And, general biology programs were very, very broad. It wasn't, and then I would look at genetics, but a lot of it was, bacterial stuff. I was like, that wasn't so interesting to me. And then literally I'm flipping through this book, and there was a section where it said plant breeding. Never heard the term, but somehow it was like, this is it. This is the term I've been looking for. I didn't even have the words for it, but here it is. And I saw that thing, and it had a number of the, major plant breeding programs in the US listed there. So immediately I was like, oh, there are some universities where they do it. They're pretty much all land grants. So like, oh, maybe I started in the wrong place, but I can correct mid-course. And it just so happened that summer I put in a job application for a USDA, temporary summer help with the Beltsville Agriculture Research Center, which is in Maryland. So that's kind of like the main USDA ARS research area. And again, this was in the days where you filled out a paper application and you mailed it in, and it went into a stack of papers, and you just had no idea what happened. But there was a soybean geneticist there named Tom Devine, who always hired undergraduates in the summer. And he somehow pulled my application out of the stack, called me on the landline, and said, we're looking for summer hires. And I think I had actually written something like, it was a government form, but you could write like what you were interested in. And I wrote plant breeding. He said, I see that you said you were interested in plant breeding. And I said, yes. Oh, of course, I didn't know what I was talking about. He said, well, that's great, because I've never met anyone who was an undergrad who actually said that. So I said, well, that's great, because I never actually met a plant breeder. So he hired me. I worked for him for 2 summers. We did, soybean genetics, a little bit of specialty soybean breeding. He was developing very small scale program, but actually that's a really good place to learn. He was developing forage type soybeans, and he then later branched out into like edamame. So these are, kind of very niche things, but they were the sorts of things where someone who had a relatively small program could maybe make a useful contribution. And then he had his like academic research on genetics, linkage mapping, stuff like that. And I was really into that stuff. So it was a great couple of summers, and it was great. I mean, it was like, yeah, we were out in the summer pollinating, it's hot and dirty and all that stuff. But I was really happy with all that. He was an Iowa State graduate himself. So he said, oh, you have to go to Iowa State. You have to go to Iowa State. So again, typing out letters on paper, mailing them off to people randomly. I mailed off to some faculty at Iowa State, University of Wisconsin, University of Missouri at that time. I think those were the 3 places I sort of checked out. And I went and I visited Iowa State and I visited Madison. It was January of my senior year. Yeah. And I was coming from Baltimore, Maryland, and, which was, I thought, cold. And then, oh, wow. And, so I said, wow, I'm going to have to make some adjustments to survive this. But it was nice. It was really good to meet the professors there. I ended up going to Wisconsin for my master's degree. I worked on alfalfa breeding and genetics with a guy called Edwin Bingham. And then I went to NC State for my PhD with Major Goodman. That came about. I sort of had the idea that maybe I was gonna, because I didn't know anything. I knew I didn't know anything. I thought maybe I should try a couple different places. So I did a master's, and it's kind of my goal. I'm gonna do a master's in one place and a PhD somewhere else. So when I was finishing my master's, I had a graduate student friend there, Martha Wilcox, and she had gotten her master's at NC State, and she went to Madison for her PhD. And she said, she's from North Carolina. She said, Jim, you have to go to North Carolina. It's so much nicer in the winter. She was right. So, I ended up coming to NC State. I worked with Major Goodman, who's the corn breeder here corn geneticist for many, many years. And now, I spent my 3 years there. That was great. We can tell stories all day about Major Goodman and his the funny things he would say. I will just quickly say, if you're a new graduate student and you're wondering if your advisor is insane, they might be. But I actually, when I first got there, I was very concerned about Major Goodman. He's a very idiosyncratic person, very, very smart. He didn't always explain things right off the bat. So I think his pedagogical technique was to sort of throw you in the deep end and see what happens. So that happened to me a few times, and it was a bit of a shock. And there was like early on, I was like, oh boy, I don't know about this. And he would, he had this very peculiar sense of humor where he when things were kind of not going well, it was sort of when he was at his happiest. And and I didn't understand that. It was hard for me to sort of fathom like how he was like thinking. But after about a semester, I stuck it out. I said, I'm not going to give up yet. I'm going to push through this. And by the end of the first semester, I had sort of realized, oh, this guy's really funny. He's actually really nice, but he's not going to show that to you right away. He's going to sort of give you this front that he's this tough guy, but actually he was he's like a cuddly bear. He was very intimidating at first, but he has a very, very good heart. So that was a great place. I learned a lot. I mean, I learned a tremendous amount, and I didn't know how to pollinate corn, for example. Like most of the graduate students I meet, they have all these years of experience where they worked in a university lab, or they've interned at a company. They know all this stuff. I didn't know anything. Like I was completely ignorant. But Major helped me. He beat back some of my ignorance for a while, and so that was very helpful for me. I did finally end up at Iowa State. I got a job there as an assistant professor, as an oat breeder. So I was there for almost 5 years, and that was great. It was a great place to work. It is cold in the winter in Iowa, and so there was an opportunity came up later at USDA at NC State, and it would be working in maize, so it would be sort of collaborating with Major Goodman, and it was a very hard decision to make, but it was a big draw to say, I can go back and work with this person who's kind of like one of my heroes. That would be amazing to be able to do that, and have a different environment. So here's my pitch for North Carolina for the people in the Midwest who are listening. If you haven't been here, you need to come. Like, we can go to the beach in about 2.5 hours from Raleigh. We can go to the mountains in about 3 hours. Now, the problem is I'm a corn breeder, so all summer I never go to the beach. I never go to the mountains. I'm in the cornfield all the time. So it doesn't really work out like you had hoped, but there is at least the potential you can get to a beach or a mountain much more quickly from where we are. So anyway, I came back here. That was 1999, and then I've been here ever since. I mean, it's funny you say I can tell people about my successful career. I'm not fully convinced. I'm not at all convinced it's totally successful, but it's been, I feel very, very lucky that I've been able to, work in an area that I really enjoy. It's the research part of this job is just incredibly fun. And as I said, USDA, they're kind of their level of direction to me is corn, improve it through breeding and genetics. And that's cool. I'm down with that. And but all the rest is sort of, I get to make decisions on exactly what projects we're working on, how we do it. It's very it's very creative in that sense, and there's a lot of scope for chasing down interesting things. Maybe when Charlie was here, I was near the peak of chasing too many things down too many rabbit holes. That's something that came with experience, but because early on, you don't know what's going to work. So I was trying lots and lots and lots of stuff, but it did get out of hand. And now that I'm entering into old man phase, my old man wisdom is like, pick a few things and focus, make some priorities, focus on those and try to do those things well. So that's kind of in the last 3 or 4 years, I'm trying to focus.
So I got to ask, I'm going to date myself here. So yeah, it's been 10 years since I was part of your program. So a full decade, actually this year has passed, and I recall, a lot of wild stuff at the time. Are we still playing around with perennialized maize? I remember that was a thing you had just started about the time I wrapped up.
Yeah, right. It's still, it went on ice for a little while. It's actually having a renaissance.
Okay.
The last couple summers. And sometimes this happens where I put some effort into it, and then I realized, well, this is actually going to be super hard. As part of my prioritization, I said, okay, this is going to go sit in the cold room. But things evolve, and my good collaborator Ed Buckler is now very interested in modifying some of the basic physiological processes in maize. And one of the ways we might be able to do that is by looking at what happens in like a perennial relative. Probably has a fundamentally different physiology when it's trying to be perennial. Understanding those physiological processes or adaptations might be informative for the sorts of things he's trying to do with the work he's doing now. So actually, Ed was the one who said, hey, you still have some of that diploperennis derived, Zea diploperennis derived material. So yeah, we pulled it back out and we're messing around with it still. I'm going to tell you, I don't think I'm going to have a perennial hybrid on the market to compete with you guys before I retire, but that's okay. It's fine. My timelines have now shifted and making baby steps is good enough in some cases. So yeah, there's a lot of I've always tried to have like a portfolio of like, here's some like real applied breeding that high probability of success is, as long as we don't do something obviously stupid. then there's some sort of intermediate things that are maybe a little more risky, but a little more novel. Those are good for graduate student projects. You know, they're probably not going to completely flame out, and they might turn into something useful. And if nothing else, it's sort of a good educational opportunity for a student. And then there's like the crazy stuff at the other end of the spectrum. And I kind of don't want to get students involved in that because I'm afraid I will ruin careers. Like my career's already, you know, it's done. It's so I can mess around with the crazy stuff. And then if something comes out of the crazy stuff, then it can, maybe that moves up into the intermediate category. You know, it might actually turn into a project for somebody.
So we've kind of scratched the surface here a little bit. So maybe I'll keep going down this rabbit hole, you know. So maybe to contrast a little bit, just, you know, being a graduate student for you versus what my experience in private industry has been since, is that, you know, chasing some of this crazy stuff that you can do in academia, you know, certainly it's one thing. I'd say, you know, my role as an industry scientist, I can do that to a little bit, but I still need to demonstrate, you know, some direct application to our product pipeline, right? You know, eventually I'm going to get my hands slapped enough if I'm spending research dollars that aren't contributing to a soybean variety for central Indiana. So, you know, maybe just some of the stuff you're working on, you know, is there anything else aside from this perennialized maize? Like, you know, you said you've got a lot of just corn improvement is your guidelines that's set by the USDA. Like, what types of things do you have going on that you can talk about that you're excited about, and how you're contributing to corn improvement now?
Yeah, so the question is, will these contribute to corn improvement? I don't know. Not, but they're interesting to me. So kind of now my criterion is there a reasonable chance something long-term might come out of it? If the answer is yes, and I could maybe justify it for a USDA research project, that's good enough. And again, I think, you know, the really fundamental difference in the world of corn breeding, which is not true for all the other species, but it's particularly true for corn breeding, is that I'm not in the business of developing products. In fact, there's no point in me even trying, because the scale of what we can do, our germplasm resources and so forth, are you know, it's just infinitesimally probable that we could develop a hybrid that would compete on the commercial market. So in that sense, again, our timelines push back then. The idea is not to make a product in, you know, industry folks, maybe it's a 5-year timeline or shorter even, where, you know, you got to get something out the door that you can sell to farmers. That's going to be an improvement, because that's why you're there. In my case, kind of in corn breeding, industry basically has the commercial sector covered. There's really no point in me trying to imitate a commercial breeding program. And so, yeah, a lot of what I do is much more like very targeted. So, you know, like Charlie worked on Fusarium ear rot resistance, only because we saw that as an area where, you know, there's some industry work on it, but, you know, it's nobody's like #1 thing to target. So maybe if we can develop some new germplasm resources or some understanding of the genetics, that would help commercial people, you know, improve that resistance trait better. So, yeah, so the Fusarium ear rot work still goes on.
Okay.
We actually we had a nice study where we wanted to see how well we could implement genomic selection, kind of rapid recurrent genomic selection versus what we were doing basically in your time, which was sort of a 2 years per cycle of S1 recurrent selection. You know, technical terms don't really matter. The question was, could we implement genomic selection with our resources, with no additional people, et cetera? Because, you know, the academic work in genomic selection is basically, here's some data, we can make predictions, they work. And we know it works. Industry has implemented this and is implementing this, they make it work at industrial scale. But then if you ask the question, well, what about like a university breeding program? Is it like cost effective for people like me to do this in a normal way? that wasn't really clear at the time. And in some cases, it's still not totally clear. Only because I'm like one guy, I have a lab technician from the USDA, I have a field technician. None of us are experts in this. So the field technician is good at like doing field experiments. The lab technician's good at, you know, some genotyping skills. But to pull off a genomic selection program, you actually have to put a lot of pieces together. And, you know, in industry, my understanding is this is all, you have teams and you have all these experts. You have like a team that just works on the genotyping. You have a team that works on the field evaluations, experimental design. You have a team that just does the genetic analysis, generates the predictions and things like that. So like, can we DIY this thing basically and do it on like a pretty limited budget? So anyway, we tried that. We basically did what we were doing before, tried DIYing genomic selection. Basically what I learned was like, we could have done it a lot better, but even kind of doing it shoestring budget style, it was effective. I mean, it actually works. As you know, that's no surprise to anyone. And I think that the idea there is that, okay, even kind of like doing this the first time, not really knowing what we're doing, clearly we can do better. Like we can optimize genomic selection even in our own hands. Whereas the phenotypic selection, we kind of already optimized it out. Like I don't know that we can't scale it up. We can't really do it any better than we're doing. So that was a nice study for me to sort of think through the whole process of like, well, okay, maybe even, you know, depending on the trait, depending on the germplasm, sometimes genomic selection is going to be more effective or less effective compared to what we can do normally. But I think you can see that the potential to improve that is only going to get better. So, you know, that was, you know, for me, it was just kind of a learning exercise to see how we could pull it off ourselves. But you know, that spun off, there was a few PhD projects for people that came out of that. So I mean, that's like a pretty applied thing. And then on the less applied side, yeah, we're messing around with the perennial relatives. I have some almost pure genetics projects going on, working on the pollen silk incompatibilities that exist in maize and some of the wild relatives, that there are some types of corn that basically can't be pollinated by other types of corn. There are actually these kinds of cross incompatibilities, and understanding the genetics of how those work has been, you know, really interesting. A lot of other people have done great work in this area. We're kind of messing around with it a little bit, but it's been a really interesting area to work in. We have, you know, we work a lot with diversity. And one of the things that happens when you're working with diversity is like weird stuff pops out like all the time. And my breeder hat is like, throw it out, throw it out, throw it out, throw it out. But then I have the other half of me that's like the genetics, and well, that's kind of interesting. So there are all these kinds of spin-off projects of like kind of weird phenotypes we've seen popping up coming out of breeding material, but I doubt you'd see them if you're working in kind of elite adapted germplasm. So then, you know, you start with a weird phenotype, then we can just let's do some genetics. Let's map it. Is it single gene? Is it a couple genes? Can we get down to identifying genes? And then maybe in the long run, the idea is that if you know the genes, maybe not me, but maybe somebody else can manipulate the genetics or the pathways to, you know, change those traits in commercial germplasm. And then the last thing I'll say is like, we've started going a lot, again, thinking through this idea that, you know, I'm not like USDA seed company. I'm a USDA researcher. And I need to be doing things that, you know, maybe are complementary to what industry is doing, but are like, they probably need to be different. They need to be more basic. So like this summer, we have an evaluation of all the open pollinated varieties from the USDA plant introduction collection, essentially the seed bank, that are from the US and Canada. So there's about 1,000 of these things, and nobody's ever systematically looked at them. So, you know, one of my thoughts was like, well, what's in there? You know, we you can see the names. Sometimes the names are informative, sometimes they're not. Sometimes the histories have been lost. We just like to know what are the phenotypes of these things. So we're growing them out in the field. We're going to measure all kinds of traits on them and just see how they look compared to like modern hybrids. Is there anything useful in there? I don't know, but we'll never know if we don't look. And we'll do some genetic analysis and try to understand the relationships among those things. And then also, besides sort of that being, complementary work that maybe industry maybe find some small piece of it useful to build on, there's sort of these niche markets of, there are growers who are interested in culinary corns for, in the South, people like their grits. You have people making tortillas, and they don't really want a typical commercial feed corn hybrid. They're often looking for something different colors, different kernel textures and things like that. And there's all that kind of variation is there in the seed bank. And so we'd like to get that material out in the field, evaluate what's in there, and say, in our environment, what varieties might actually be good if you're like a specialty grower and you want to try one of these old varieties. Because some of them are hard to grow and some of them are really unadapted. So you don't want to burn everybody's enthusiasm by having them grow some really unadapted, difficult material. But there might be something in that collection that would be more immediately useful. And then on top of that, we have a little bit of, we got some recurrent selection going where we're doing basically within open pollinated variety improvement, pretty much like what people were doing in the 1940s and 1950s. So there's nothing new here, but it's targeted really at these specialty food types for culinary use.
I got to ask, because I know it existed when I was there. Is there a Jim Holland brand elote street corn that I can find yet? Have we gotten anything out of that project yet?
Not quite, but I think it might happen. It could happen.
Okay.
So I haven't given up on it.
Lance, you tried coming off mute a minute ago. Did you have a question?
Yeah, I have a lot of questions that I could ask you.
Yeah, certainly. It doesn't have to be just me driving this, man.
Yeah. Well, since you brought up, I was kind of interested in some more technical questions. You brought up recurrent selection, right? And something I've been very interested in since beginning of my grad school, just because, you see very little academic papers really on the actual use of recurrent rapid selection using genomic selection. So I just wanted to ask you about how you kind of went about it. Did you try and set up like multiple cycles of selection through a year, or did you just integrate it into your general breeding program where you just, once a year you made selections based off of predictions and then went on that way?
Yeah, basically we integrated it with the regular program. I don't really have a greenhouse assigned to me, so I didn't have a greenhouse. I did use winter nursery though. And the advantage there was we were genotyping individual plants in the winter nursery and intermating. In some cases we self, but in some cases we just directly intermated, and basically implemented using the genomic predictions in the off-season nursery, where normally there's no way we were going to get good phenotype data from those. So that was the idea, was that genomic selection was going to give us that second environment. And the other thing, and Charlie can tell you this, is that single plant phenotypes for this ear rot disease are almost worthless. There's so much plant-to-plant variation, even within like a uniform hybrid row or a uniform inbred row. There's a lot of environmental effects. So you need a lot of reps on the phenotypes before it means anything. And the advantage of the genomic selection was to train the model on good, highly replicated data where we had good heritabilities on the thing. And then we could go to this single plant selection method. And it basically works, right? So that was good.
Did you do predictions on mainly that ear rot then?
That's all we did. Yeah.
Okay.
Well, okay. Ear rot and the mycotoxin accumulation, which they're closely they're highly genetically correlated traits, but we measured them both in the training set. So we predicted them both and we made a little index on it. But yeah, it was very, I mean, this is why it's very academic, because we were like, we're just going to focus on this disease. Because I don't want to spend 20 cycles or 10 cycles. I did it in like basically 4 cycles. And we wanted to see some impact pretty quickly. But again, that's the difference between an academic group and, you know, an industry group is saying, well, you could do genomic selection for one trait, but, you know, that's not a product at the end of that.
And is that you're right, extremely heritable as well, like most other diseases? Or is it more of a quantitative resistance?
It's highly quantitative. There's no immunity. There's no major genes. Charlie GWAS'd it. He was the first person in the world to do a GWAS. He hit some things, but the effects are really so small that, you know, the idea was like, okay, Charlie, you're going to find some like moderate effect resistance genes that we've so far failed to find. And then we can do targeted marker selection on them. And, you know, Charlie identified some genes, but the effects were too small to really be of practical use for breeding. So it's highly quantitative, and that's where, like, when, you know, then you start reading the literature, oh, this is what genomic selection's good at. You don't need a particular marker. You just you're looking at the whole genome because it's polygenic. You know, it kind of works that way. Is it heritable? It's heritable if you replicate highly. Okay, on a heritable in the sense of like an animal breeder where every animal is the unit of selection. No, it's not. It's almost not heritable. But if you have replicated progenies, it's moderately to decently heritable. But to make it heritable, Charlie can tell you what it takes. You know, walking through the field with the backpack full of growing up the fungus, making the inoculum, walking through the field, poking the ears of the corn and shooting this inoculum into them, harvesting everything by hand, rating everything by hand. If you want to get the mycotoxin, then you have to shell everything, grind it, do an ELISA assay, which is expensive, would take the grad students all winter to do this sort of thing. So yes, it's heritable if you can put all that effort into it.
I have a lot of core memories of grinding corn at Reedy Creek, North Carolina. And as a personal note here, I know I have switched crops since, but I have not gotten away from Fusarium. One of the main focuses of my breeding program now is sudden death syndrome. We run a screening nursery at Windfall, which is coincidentally also a Fusarium species. So I've built my entire career around that genus.
Well, Jim, since you brought it up also, and that is, you brought up GWAS, and I mean, that's where I first saw your name as a grad student. Some of your collaboration with Buckler's lab and things like that. So, you know, when a lot of people cite original papers, it's a lot of that originally unified mixed model that you were on. So I was just kind of asking, what was your involvement in some of that really early GWAS applications and things like that?
Yeah, that's a great question. You know, I would not claim a major contribution to it. That original unified mixed model paper is really the work of Jianming Yu, who's now a professor at Iowa State, you know, along with Ed Buckler and some of the other folks there. You know, my big contribution was Jianming came to visit me for a day or two because I knew a little bit about mixed models then, and it was really, you know, there weren't many plant breeding people who were doing much mixed models analysis work, and I just happened to be one of them. So Jianming and I, you know, he came and we kind of talked through some mixed models issues on, you know, how you could implement these tests. But it's not, I'm not going to claim it wasn't like my idea. Like it was sort of like Jianming showing me what he's doing, and I'm like, wow, that's cool. That's a great idea. That was my contribution. So a lot of, you know, there's a lot of stuff too where, like, yeah, it is funny is like, I'm kind of getting into an old man phase, but it's like I will meet graduate students who are like, oh, you know, you did all this work, and I'm like, I'm on a lot of papers, but I didn't. Charlie can tell you, I don't really do all the work. I show up, drink my coffee, talk to people, tell some jokes. We write some papers, and other people are doing the real work in many cases.
You kind of mentioned already, and you've name dropped Ed Buckler a couple times. And as I look through, some of the stock interview questions that Lance had sent me before this, collaboration was something that come up with. And I know industry, a lot of times we have a lot of internal collaborations, external, we're working towards that, can be a bit more limited. But what does that look like for you? I know you talked already about how you're doing a lot of stuff DIY in-house, but what other universities, like who are some other folks that like you work with and that you enjoy working with and collaborating with, who are your peers?
Yeah, it's a lot. And actually that's the fun part of it is like, I get to work with my friends, even if they're not here, they're at other places, and that it's really enjoyable. So, for example, the heirloom variety evaluations we're doing, that's in collaboration with Sherry Flint Garcia at the University of Missouri. Actually, sorry, she's USDA ARS, located at the University of Missouri. So and I've known Sherry from when she was a postdoc with Ed Buckler, when Ed Buckler was at NC State, many years ago. So we go back a long ways, and it's just, she's for me like a great person to just talk to. I think she and I share the enthusiasm for kind of looking at weird corn, and it's nice to not be the only one. So that's a great one. And I'll also say here at NC State, I have a lot of great collaborations. So Peter Ballant-Curdy, who's a USDA maize disease genetic specialist. I mean, I've worked almost the whole time I've been here. I've done a lot of work with him, and he's a great scientist, a really good person to work with. Joe Gage is a new professor of genomics, and he's just like amazing to work with. He knows all this stuff that I kind of heard about. I don't really understand any of it, and so when I have questions, I go to Joe, and he patiently explains things to me. And then I also, we have Matt Kurkowski, who runs the germplasm enhancement of maize program, which is that's a very applied, one of the few applied public corn breeding programs. So it's good to, I enjoy that in our setting, to have the collaborations that run from the really applied breeding to the more basic genomics and genetics. That's really nice. I'm doing a lot of collaboration, so I don't want to leave anyone out, but we're working on some grain protein work with Candy Hirsch, also with Sherry again, and Paul Scott at Iowa State. So that's some new stuff that's coming up. I have another relatively new colleague here, Ruben Rayan Alvarez, who's in biochemistry, but he's really like a maize diversity genetics person. He's at NC State. He's great to work with. Yeah, I don't know. I'm gonna leave people out, so I'll stop. But I'm still collaborating a little bit with Ed Buckler, which is always like incredible. He's just like ideas just like flying out of his head. That it's very inspiring to work with people like that.
And you're still, I assume you still have a full complement of graduate students at any given time, right? As far as your education commitments for training graduate students?
Yeah. I mean, one thing that's changed since you were here is that there has been some little bit of rule changes. So like, we have to be co-advisors. We have to have an NC State faculty member co-advise.
Okay.
To make sure that the USDA is not taking over a university role. So that's fine. But like I said, here I have a lot of good collaborators. I have at least half of 3 graduate students under my wing right now. Yeah, and it's fun, but that's amazing too. So I have one graduate student who's funded by FFAR, which is that's a funding mechanism that has a connection between academia and industry. So actually she's doing the actual lab work at Syngenta, working with their gene editing group and maize. So again, that's stuff that there's no way we could do that here on our own. I cannot DIY gene editing. Although I will say that actually the public resources for that are now much, much better than they were when we started this. So you can actually basically pick a target gene and talk to the people at the University of Wisconsin, for example, and get an edited knockout. But anyway, so I have one student working largely at Syngenta to do their research. I have another student who's carried on the fusarium work and also, branched off into some genomics for his PhD. And then we have a brand new student who we came in on the grant funding we have for this heirloom work. And so, yeah, I'm trying to keep it small because I can only manage so much. And like I said, I'm trying to focus, trying to put things on the priority list, and the things high on the priority list should get a graduate student assigned to them. And then, if it's too risky for a graduate student, I probably should not be growing 1,500 plots of it in the nursery. That's like, I've learned, it's taken me a while. So yeah, there's graduate students. I have a USDA postdoc right now. I also co-advise a postdoc at NC State working on genomes to fields. Oh, I forgot. Yeah, I collaborate with the genomes to fields crowd. So that's, Natalia De Leon, Sean Kepler at the University of Wisconsin. They're like really leading the charge there, but they're super fun to work with, and it's all remote. That's a modern thing where we can just have Zoom meetings once in a while and email stuff back and forth, and so far it's worked really well. It's been very enjoyable. So yeah, I'm still trying not to ruin careers, but try to be a positive influence on a few people at a time.
Well, and I mean, part of the reason I asked that too is, and again, as I give a compliment to you, that for listeners who may not be as aware, when I think of, NC State and your capacity in the USDA at NC State, that institution, I would argue, has had an outsized influence, at least on what I see in private industry. Just the number of corn breeders that, let's say, either came from Major Goodman, yourself, or people he trained directly, and then people that his progeny have since trained. So, Lance, you had Dave Bubeck on recently. He came through the NC State pipeline. Again, just to speak for Corteva and Legacy Pioneer Hybrid, our director of plant breeding, Jeff Graham, was a Charlie Stuber progeny, right? So there's, I can't understate the importance that, the work you do and Major did before you to, the impact it has really had on putting plant breeders on the path they are on today. So, yeah.
That's very kind.
Lance,
what I tell my students, this is the last, sorry, I have a lot of dad jokes and old man.
No, you're good.
Be patient with me. But one of the things I have regularly told my students is, I cannot teach you how to be a plant breeder, right? I can teach you how to not be a plant breeder, because I've done everything wrong, and I continue to find new ways to do things wrong. And so just listen to me when I say this is not going to work, because I've tried it and it doesn't work. And I can't tell you what to do, but do something else. So I feel like that's my contribution to the education of future plant breeders.
So
don't be like me.
So Lance, I know we're getting close to our time here, so maybe I'll ask one last question here and kind of end on a fun note. And I have no idea what your answer would be this to be, but I saw one of the last questions that was on the script here was something on just a fun historical story. And when we started this conversation in your career, you talked about your time with Major Goodman, and I remember Major was still around. I was in graduate school, and yeah, some of the cynical stories he would tell me. Give me one story, a fond memory you have of either something that shaped you or just something that was classic Major that you can give to the rest of the audience here, and how it impacted you.
Well, maybe I'll tell you near the end of my time there. So this was after I understood his humor. I interviewed for this professor job at Iowa State. So it's Iowa State, right? This is like a big deal. I get a phone call from the department head who says, "We're going to offer you this job." So my jaw's on the ground. I can't believe this. They're offering me this job. This is so crazy. So anyway, I'm excited. I'm all happy. So I hang up the phone and I walk to the next room where Major's sitting in front of the computer analyzing some data. And I say, "Major," I said, "Iowa State just called." I said, "They're going to offer me that professor position." He looks at me and he says, they must be desperate. And again, if it had been week one when I didn't understand who this guy was, I would have said, what's wrong with this person? But I had known him for 3 years at that point, and it just made me laugh so hard. I was like, okay, there's Major, summed up perfectly. Yeah.
Yeah.
But the thing about Major was he would make those comments, but he always reserved the most disparaging comments for himself. So his classic lines were, my lines are just junk, and then I cross trash by trash, and I get more trash, and I took some of your trash, I crossed it with my trash, there's more trash, so that was, he would never brag. Let's just say he did always the opposite of bragging, telling you all the problems with things rather than accentuating the positive. So that was always sort of the fun thing. So when Major told you something was terrible or bad, you had to like rescale it, because it was like bad on the Major scale sometimes could be really good on, a normal person scale.
Well, good. So Lance, maybe with that, I think we've gone probably about 45, 50 minutes here. Do you have any comments here to wrap us up, Lance?
Yeah, well, I just want to say thank you, Jim, and thank you, Charlie, for talking with us today. It's been a very, very interesting 40, 50 minutes. So I really appreciate that. It's been pretty
Talk
fun.
a lot, sorry.
Oh, no.
No, I appreciate it. That was the goal of this, was to find out who Jim Holland is.
Yeah. But with that, I just want to say thank you again, and thank you for everyone who's listening. And I hope you enjoyed today's Breeding Bites podcast. So thank you.
Yep.
Thank you.
Thank you.
Bye.